Our presentation today is How Can Model-Informed Drug Development Accelerate Your Program? Our speakers are Dr. Krithika Shetty and Dr. Miriam Traim. Krithika is currently a Director of Clinical Pharmacology at Certara. Prior to coming to Certara, she was an oncology clinical pharmacology reviewer at the FDA. She holds a degree in pharmacy from the Birla Institute of Technology and Science in India and a PhD in pharmaceutical sciences from the State University of New York in Buffalo. Joining Krithika is Miriam, who is currently a Vice President of Pharmacometrics at Certara, where she leads the United States Northeast Regional Lead for Quantitative Science Solutions. Miriam is an expert in cell and gene therapy, and she has more than 12 years of industry and academic experience in clinical pharmacology, pharmacometrics, and systems pharmacology. Welcome to the presentation. Krithika, the floor is yours.
Good morning, everyone. Thank you for taking the time to attend. I'm happy to be here today and to be speaking with you about the role of MIDD in clinical pharmacology regulatory decision-making, as well as its contributions to smoothing the drug development path to initial drug approval and beyond. Next slide, please. Next slide. Next slide. Excellent. So as many of you are no doubt aware or have experienced, the FDA has a long history of engaging with drug developers in order to increase the utilization and adoption of MIDD-related approaches in regulatory decision-making and drug development. The FDA routinely evaluates new MIDD approaches as well as existing applications of MIDD approaches to improve their utility in the overall drug development pathway. At the FDA, MIDD approaches are typically viewed as a means to integrate the totality of evidence in front of the FDA. So it's a platform to integrate evidence related to the current understanding of a disease state, drug pharmacology, as well as patient-related factors in order to make informed decisions about key elements in the drug development program, such as dose optimization, clinical trial design, as well as overall risk-benefit decision-making. Ultimately, clinical data that's developed, generated as part of a clinical development program is integrated with MIDD-related information and rolled up into recommendations in the US prescribing information. Next slide, please. This slide, which most of us are no doubt already familiar with, summarizes the utility or the applications of MIDD in various stages of drug development. Early and non-clinical development approaches such as PBPK or PBPK-PD, QSP, and PK-PD modeling are used to aid decisions such as the selection of the target, the drug target, or lead candidates, to assess the viability of a program or a candidate in the context of a therapeutic product profile, and ultimately to translate non-clinical toxicology and pharmacology information to humans in order to inform first-in-human dose predictions. As development proceeds through phase 1 clinical trials and initial human data starts to become available, approaches such as QSP, PK-PD modeling, preliminary population PK or exposure response analyses are used to guide decisions such as dose escalation, selection of dose regimens for proof of concept, proof of mechanism, phase 2 type studies, as well as to make early go-no-go decisions on programs. This process continues iteratively through phase 3 development, where modeling approaches are used to inform recommended dosage for phase 3 studies, as well as to inform dose modifications for special populations such as those with organ impairment. And ultimately, ahead of a marketing application submission, all of this information is integrated together in order to inform key elements of the submission and the USPI, such as recommended proposed dose for the label, and dose modifications for special populations. While this graphic ends here, certainly the utility of MIDD in drug development does not, with applications in the post-market setting, including pediatric programs, new indications, as well as regimen modifications. Next slide, please. In order to exemplify some of the notes we've just been discussing on the previous slide, slides. This slide provides an overview of the question-based review format that the FDA currently uses. As you can see, the QBR review template requires reviewers to address certain key questions which benefit from, if not require, the use of MIDD to fully address. And this includes whether or not the clinical pharmacology program provides supportive evidence of effectiveness, whether or not the proposed recommended dosing regimen is appropriate for the general patient population, and whether or not special dose modifications and management strategies are required for subpopulations, including based on intrinsic factors, and whether adjustments for DDI or food-drug interactions are necessary. As you can see, PK characterization and appropriate pharmacometric analyses are key components of adequately addressing each of these questions. These approaches certainly provide evidence or supportive evidence of a drug's activity, including mechanistically supporting the adequacy of a single well-controlled trial. We use it as a central component of supporting the proposed dosing regimen, including identifying subset of patients that will either disproportionately benefit or experience toxicities of the drug, and understanding the impact of intrinsic factors on drug exposure and PD, which ultimately helps dose management strategies and dose modifications in the label. Next slide, please. As a FDA reviewer on a day-to-day basis, it was just part of my job to use MIDD analyses and approaches in order to guide my decision making. For example, I used PopPK and ER analyses to support dose justification for the general patient population, to understand and clarify trends in efficacy, including sort of anticipating losses of efficacy, understanding mechanisms of toxicity, including explanations for potentially rare toxicities. A nice example of this from my personal experience was the review for Tivdak, where despite significant concerns of toxicity at the proposed recommended dose and limited understanding of dose-response relationships at lower doses, further evaluation of alternative dosing regimens was not recommended because ER analyses suggested a decline in efficacy at lower exposures, which in the context of the observed ORR, DOR, and the late-line setting, late-line indication, was able to guide the clinical decision that such a further loss in efficacy would not be acceptable. Many of the nice examples for the use of MIDD in justifying dosing recommendations for subgroups also come from ADC reviews, where reviewers oftentimes have no choice but to rely on the totality of evidence integrated via PopPK and ER analyses in order to guide dosing regimens, specifically a close understanding of the exposure of the payload with toxicities and exposure of the ADC with efficacy. There are, of course, numerous nice examples of PBPK being used to expand labeling, sometimes without clinical studies, but perhaps the most prominent example is the use of modeling and simulation in order to extend the labeling of pembrolizumab to include the 400 mg Q6W dosing recommendation in the middle of the COVID pandemic when the effort was to keep patients out of the clinic. By and large, I think generally speaking, good PK data and pharmacometric analysis may be used to diminish the need for post-marketing studies by sort of forecasting the utility of these types of analysis. In the absence of good PK data and appropriate analyses, reviewers have no basis on which to complete a risk assessment, and therefore are far more likely to request dedicated studies in the post-marketing setting or to make more restrictive amendments to the label language. Next slide, please. The utility of MIDD approaches, especially in the oncology context, is only expected to go up in the next few months and years with the launch of Project Optimus, which is spearheaded by the Oncology Center for Excellence at the FDA and jointly led by both clinical as well as clinical pharmacology review divisions at the FDA. Project Optimus is aimed at reforming dose optimization in oncology in order to maximize not just the efficacy, but also the safety and tolerability at the recommended dose. This program applies to all new drugs and biologics being reviewed under CDER Oncology and has a strong emphasis on understanding dose and exposure response relationships of the therapeutic, as well as the therapeutic window, which requires a good characterization of the PK and PD profiles of the drug. Next slide, please. So under Project Optimus, the FDA is looking at 4 or 5 key areas to provide supportive evidence of appropriateness of the proposed dose. These include translational data, clinical PK, clinical PD, clinical efficacy, and safety information. As you can see from the highlights on this slide, fully addressing each of these areas would require, if not benefit from, a good application or good and early application of MIDD approaches, which includes elements such as dose PK relationships, both in non-clinical species as well as in the clinic, understanding of dose exposure response relationships in non-clinical species and translating this to the clinical setting, and ultimately being able to understand, as development proceeds, exposure dose response relationships with clinical safety and efficacy endpoints. Next slide, please. The launch of Project Optimus brings us to an interesting junction where there's an opportunity to expand the toolbox of MIDD approaches that are typically used for dose optimization, not just in oncology, but in other indications as well. The box on the left summarizes some of the more well-socialized or better-known approaches that everybody is comfortable and familiar with, which includes PK/PD models, PBPK or PBPK/PD models, population PK analyses, and exposure response analyses. The box on the right justifies some or summarizes some approaches that are currently still underutilized but are increasing in their adoption by drug developers, and have various degrees of regulatory experience and comfort. And these include things like clinical utility index analyses, longitudinal modeling, receiver operating characteristics or ROC analyses, clinical trial simulations, the use of KPD models in situations where PK data might be limited, as well as, importantly, the increasing adoption of quantitative systems pharmacology or QSP models. Next slide, please. A few applications of MIDD-based approaches under Project Optimus and other therapeutic areas for that matter include translational or pharmacological target concentrations from non-clinical species to the clinic in order to inform early escalation and initiation of backfill or expansion. The prediction of target site exposure and pharmacodynamics for the various active drug moieties of species, an early understanding of sources of variability in exposure, as well as exposure response relationships or predictors of clinical response. Approaches such as longitudinal modeling enables sponsors to better leverage clinical data when there's significant dose modification as part of early trials. CUI analyses permit integration of both safety and efficacy data to better understand risk-benefit balance across a range of doses, as well as to maximize the use of various PD endpoints when they have unknown relationships to clinical activity. ROC analyses permit at least attempting to define optimal concentration cut points associated with various efficacy or safety metrics. And of course, QSP has been used extensively in the past few months and years to narrow down search space for things like priming dosing regimens for T-cell engaging bispecifics, which have CRS risk, to support program design, as well as for dose range selection for new indications and combinations. Next slide, please. I'd like to sort of close my portion of the talk with a couple of slides that help to solidify or provide more context for the role of MIDD in the future of the FDA and drug development. This slide summarizes the MIDD paired meeting program. As of 2018, under PDUFA 6, the FDA launched an MIDD pilot meeting program, which permitted drug developers to speak with, to engage early with the FDA to discuss the application of MIDD in medical product development. As you can see from the figure on the right, this program has had robust uptake among sponsors, not just in oncology, but across a variety of different therapeutic areas, and has spanned discussions ranging from first-in-human dose selection all the way up to phase 3 trial design and post-marketing questions in that context. As a PDUFA 7, this is no longer a pilot program. PDUFA 7 made the MIDD paired meeting program a formal program, which, as I said, certainly helps to solidify the importance of MIDD in decision making at the FDA and in the future of drug development. Next slide, please. And just to close, I'd like to share this graphic from the FDA Office of Clinical Pharmacology 2022 Annual Report, which nicely demonstrates how MIDD is one of the central focus areas of research at the FDA. What is implicit in this graphic is not only are there several MIDD-centric or MIDD-focused projects, but also MIDD also finds a way to trickle into the other research focus areas at the FDA, including DEI related topics, as well as safety and toxicity. In summary, I think it's fair to say that MIDD will continue to be a key driver of innovation at the agency as time proceeds. The current trends that we observe will only strengthen, and the influence of MIDD is only expected to increase with the advent of new technologies such as AI and machine learning, with the FDA, which the FDA has been watching closely and has started to include and take up. So with that, I will thank you for your time and attention and invite Miriam to take us through her portion of the talk. Thank you.
Thank you, Kritika. Yeah, so in our second part of the webinar, we're going to talk about what is clinical pharmacometrics and how it can actually be utilized to accelerate your clinical programs. So pharmacometrics, we are using in silico mathematical models to understand the drug-patient disease relationships and then to optimize the drug therapy through our dose exposure response analysis. So pharmacometrics can really help us with understanding the potential that compounds offer to patients by optimizing the dose regimen, the time courses of treatment, as well as drug individualization, while we can still account for the disease progression. And so then we can use our models to support clinical trial designs, program decisions, as well as regulatory submissions. Next slide, please. And why is dose finding so hard? So we are dealing with lots of variability in our patient population. We are dealing with nonlinearity sometimes in our drug concentrations. We have a lot of changes over time happening in drug concentration, biomarkers, as well as disease progression. And we also need an ability to capture and measure disease markers. Usually we have a very defined patient population in our clinical trials in which we are testing certain dose levels. And this information can then be utilized to understand our pharmacokinetics, so our concentrations over time, as well as our pharmacodynamics, which is the relationship between the effect and the concentration. And then when we put this both together, we can then predict our effect over time. Could you advance one, please? We can then utilize our models to apply them to a new population, for example, with a different genetic pool and disease, by inclusion of population-specific covariates to predict then the dose exposure response and to dose individualize and for a new population. Next slide, please. As already mentioned, our drug concentration and responses, they are wax and wane over time. And so we are dealing with a vast majority of different timescales. Advance, please. So on the PK end, we are seeing changes happening in minutes and hours. And then on the biomarker side, we are dealing with fast biomarkers, which change, for example, over days. And next would be have the slow biomarkers, which are changing. Advance, please. And advance again, please. Our slow biomarkers, which are changing over months. And then our clinical responses sometimes are not changing before years. So in our pharmacometrics analysis, we can actually take into consideration these different timescales and can sometimes predict from very early on what happens in late-stage clinical outcomes. Next slide, please. So then always the question is which model to apply. And here we need to really consider observation levels in pharmacology. As we know, disease processes can be characterized at actually different levels. Diseases can manifest on the molecular level, and it can lead to disturbances at the cell or tissue level. And this then can lead to the establishment of clinical signs and symptoms. And automatically, information from these three levels can be used to characterize and predict the clinical outcome. So when we now look into the modeling part, models based on clinical data are typically drug-centric and descriptive. And then on the other hand, when attempting to characterize all the biological processes involved in drug responses, these models are becoming typically quite complex and describe the various processes in the underlying biological network. Both approaches have advantages and limitations, and usually we have a very specific question at hand and are looking into more fit-for-purpose models, which are the mechanism-centric models, and they represent a compromise between the two. And it should be noted that these models ideally do not only focus on the benefit of the drug treatment, but also on the risk. And to maximize the benefit-risk ratio, clinically meaningful covariates, as you can see here in the picture, for example, omic data, can then be considered in order to optimize the drug dose. So these mechanism-centric models are currently used to characterize the dynamic interaction between drug, the biological system, and disease at multiple levels, evaluate the effect of combination therapy, as well as distinguish between treatment effects, also given in an appropriate study design. Next slide, please. So with this, I would like to come to our first example where the role of MIDD played a key role, and this was in the development of pembrolizumab. Next slide, please. Pembrolizumab is an anti-PD-1 monoclonal antibody which was developed by Merck et al. At the time for the treatment of various cancers. And the initial clinical development focused on melanoma only. The clinical development and initial approval very largely was based on a single study, which was the KEYNOTE-001, which was a phase 1b first-in-human study of the safety and efficacy of pembrolizumab, and first in patients with solid tumors, and then later it was also tested in patients with advanced melanoma. And the study sought to address actually the following key questions. First, what appropriate dose range was to investigate in clinical studies? No, back please. Second, what is the appropriate dose regimen that balances the risk and benefit, as well as what dose adjustments would be required due to the potential effects of intrinsic and extrinsic factors on the pembrolizumab exposure? And key to answering these questions was actually the interconnectedness of different models utilized during the development of pembrolizumab, as depicted in the right figure. So if we go through the figure on the right from top to bottom, we see that the first modeling effort involved a population PK modeling and an empirical PK/PD modeling of target engagement using interleukin-2 data from an ex vivo assay. And the goal here was to pick the minimal effective dose for future cohorts using a learn and confirm paradigm. As a second modeling effort, a translational PK/PD model of tumor growth inhibition in mice was used to support dose selection for pembrolizumab, and the goal here was to identify the minimal effective dose for pivotal efficacy and safety cohorts. But this time the model used mouse tumor data and translated these to the clinic in using human simulations. As a third modeling step, we used the traditional PopPK model, included the clinical data to investigate the effect of intrinsic and extrinsic factors on pembrolizumab pharmacokinetics, as well as to support the drug label. And then the model was utilized using pooled data from 3 studies and included patients with advanced melanoma, non-small lung cancer, as well as other tumor types. And then as a final part of the modeling, the population PK model of tumor size dynamics was utilized to quantify the efficacy and safety exposure-response relationships for pembrolizumab, as we see here, by predicting overall survival and adverse events. Next slide, please. And as a conclusion here, we can see that already from the left to the right, that already by utilizing the ex vivo interleukin-2 assay data, we could identify that the exposure associated with a complete functional blockade of PD-1 in the ex vivo assay was observed with doses of 1 milligram every 3 weeks and above. And then the PK/PD modeling concluded that the probability of achieving a full target engagement in at least 90% of the subject was achieved with a dose of 2 milligram per kilo. And then later with the PopPK model, we could actually verify using the exposure efficacy and safety analysis that also here we had no difference in tumor size changes as well as no difference in adverse event signals when we used it at 2 milligram per kilo dose every other week or 10 milligram every other week or every 3 weeks. So hence this integrated modeling approach was able to support the study design and the initially approved dose of 2 milligram per kilo every 3 weeks as well as the labeling. Next slide, please. This is another example where MIDD is playing a key role currently in the development of a bispecific antibody in pediatric cancer. And this is the odronextamab, which is a human CD20, CD3, IgG4 based bispecific antibody. And it is actually currently also in development in adults with relapse refractory CD20 positive B-cell malignancies. However, there was an immediate need to in parallel start the development in pediatrics for non-Hodgkin lymphoma. And key challenges here were to answer what is the appropriate pediatric dose regimen for ages ranging from 6 months to 18 years. We had to take into considerations differences in PK parameters in adults and pediatric patients driven by differences in body weight. We also had to think around maturation effect on the non-specific drug elimination to be considered, in addition to the weight effects for infants younger than 1 year of age, as well as around the similarity of the target and the tumor burden in adult and pediatric patients, which supports similar target-related non-drug clearance. Next slide, please. So the developed adult PK model was utilized, which included a parallel linear plus a time-varying nonlinear elimination and body weight and age on clearance and volume. Then the virtual pediatric population with demographics from the enhanced database was utilized for simulations, as well as that a maturation effect was included on the non-specific elimination in addition to the body weight effect for infants less than 1 year of age. This model was then utilized to simulate Cmax concentration of odronextamab in the first 3 weeks with a split dosing and weight tiered regimen, same as it was utilized or applied in the adult population, as well as what semen concentration was simulated of odronextamab at week 12 after weekly and at steady state after biweekly dosing. And so it was important for us to look at the maximal concentration across the different weight groups during the tiered dosing regimen in order to manage the safety risk of cytokine syndrome, as well as then to look at the minimum concentration which was required for efficacy across the different weight groups. And as you can see here on the left in the figures, for the maximal concentration as well as the minimal concentration, they are in very good alignment across the different weight groups, also compared to the reference, which is the adult one all the way to the left in each of the figures. So this then allowed to simulate a proposed fixed dose regimen by weight, as you can see in the table on the right, and this is currently evaluated in clinical trials. Next slide, please. In our last example, I would like to highlight what the role of MIDD is playing right now in the current development of cell therapy development. Here we also have the question, what is actually the optimal CAR T cell dose? And unlike conventional drugs, CAR T cells typically proliferate within the recipient, which results in a highly variable relationship between the dose and the exposure. CAR T cells are usually administered via IV infusion, which is then followed by a rapid redistribution, as you can see here in the dip of the circulating levels of the CAR T cells. And they are redistributing in the body into different tissues, including the tumor sites, within a few hours. And this is observed in this initial drop of the circulating CAR T cells. So the redistribution phase is then followed by a phase of rapid expansion of the CAR T cells due to activation and proliferation of CAR T cells. And circulating CAR T cells frequently peak within 2 weeks of administration, after which they decline at a very variable rate. So, and to measure the CAR T cell exposure, we use the maximum CAR T cell level, so at Cmax, and the area under the concentration time curve between the time of CAR T cell administration and day 28. And both Cmax and the AUC then reflect early CAR T cell expansion and are associated with clinical responses to the therapy, with higher AUC values among those who respond usually. If we go to the next slide, the reason why we're doing this is because the FDA actually reviews and evaluates the justification of the proposed to be marketed CAR T-cell dose as part of OCP and Pharmacometrics review in a very similar fashion as for small molecules and other biologics. So they are very interested in understanding the exposure response evaluations to predict the clinical outcome. And here you can see a few examples from previous submissions, from Yescarta, Tecartus, and Abecma, for example, where different types of analysis were performed. For example, logistic regression, time to event modeling, or the duration of the response using Kaplan-Meier curves. As well as to the right bottom, we see that the CRS, which is the cytokine release syndrome rate, was linked to the exposure, in this case the AUC, to really understand the relationship also on the safety side. We then also want to understand why we have so much variability. So if we go to the next slide, the next step, we would then look at key factors that can affect the cellular kinetics, because we know by now that the dose alone is not the only factor which drive expansion and the variable decay phase we see in CAR T-cell therapy. So we need to include look into intrinsic factors such as demographics, the patient characteristics. So the antigen load, tumor burden plays a big role, as well as the CAR T-cell fitness, very important, especially for the autologous treatment. Cytokines and other pharmacodynamic markers are playing a big role, as well as the immunogenicity markers. And then especially for the allogenic CAR T space, we need to look into the HLA matched versus non-matched donors. Extrinsic factors which can influence our CAR T-cell expansion and persistence phase might be the dose itself. Should it be a body weight adjusted dose or a flat dose? Other medications. So the lymphodepletion regimen plays a key role, the bridging therapy, as well as tocilizumab and steroid use during CAR T-cell treatment. We also need to think around our drug product, so the CD4 and CD8 ratio, the composition of the manufacturing parameters, as well as our CAR construct. And then we need to also understand manufacturing differences. Usually we don't necessarily manufacture at one site, so there can be huge lot-to-lot differences. So all this can be evaluated using model-informed drug development by utilizing the Pharmacometrics models. And I would like to conclude this with a last slide, how important the lymphodepletion regimen is. So this is really key to success of the CAR T-cell therapy. And usually if we are in the autologous setting, we're using cyclophosphamide and fludarabine, although also here it has been shown that adjusting the fludarabine dose is important and critical for optimal outcome of your CAR T-cell therapy. But if we now look into the allogenic space, we usually add an antibody. This antibody is added to knock out the NK cells. And this, in this case, is a repurposed entity. It has not been approved for lymphodepletion before, and also had never been tested in combination with fludarabine and cyclophosphamide. So this falls into Project Optimus, which my colleague Kritika already highlighted, and needs to be dose optimized in the same way as other drugs in oncology. So here we are applying usually an optimal design approach, which is the definite screening design, the DSDS design, where we can help to really ideally optimize the phase 1 trial to test all 3 components in the lymphodepletion regimen, plus the CAR T dose, to have an optimal design to then in the end be able to define after this phase 1 what are the optimal doses of all these 4 components to accelerate through the drug development pipeline. With this, I would like to conclude this section, and thank you for your attention.
All right, fantastic. Thank you so much, Miriam and Kritika. And we are now going to invite our audience to pose some questions to our speakers. So just looking at the questions. This might be one for Kritika. Does FDA have a specific area of interest, focus under the MIDD paired meeting program?
Right. So by and large, the program is open to all new drugs and biologics program that have an IND under review with the FDA. The website publicly announces that the focus areas of interest include questions around, or MIDD approaches used to help address questions around dose optimization, clinical trial design, including sort of just overall selection of endpoints and study design, as well as a prediction of outcomes, as well as a more mechanistic understanding of safety endpoints and outcomes as part of the overall drug development program. What the FDA explicitly says is excluded as part of the MIDD paired meeting program is very detailed statistical analyses and related questions, adaptations of the above. So those will have to go through separate channels. Yes, I hope that addresses the question.
I think it does. Thank you. Looks like we got a good number of questions coming in. Question for Miriam. The PK-PD relationship is really hard to display in many cases and indications. Is there any clear path for demonstrating the PK-PD relationship or any surrogate or replacement?
So I'm not quite sure what is meant with the last part, but it's true of the question about the replacement. Maybe that whoever posted this could please clarify the question there. But yes, it's not always easy. We don't always have a good biomarker, but if we do have a good biomarker, it would be really helpful to utilize this biomarker and make use of this.
Okay, fantastic. Someone wants to know if you could comment a little bit more about the role of exposure response analysis in answering some of these questions.
So exposure response analysis, we can implement this usually very early, right? We can use our preclinical data already and do a dose exposure response. We don't necessarily need any exposure. So if we do the dose efficacy or dose modeling, for example, using the mouse tumor data, we can already predict very well, usually, what is the efficacious dose range in the human study.
Thank you, Miriam. Krithika, this might be a good one for you. Someone wants you to talk a little bit about some of the limitations of these different approaches, PBPK versus PopBK versus QSP.
So as with everything, this is a true conform answer. It's context specific, right? So different approaches will have different levels of utility, regulatory comfort, and success, really, depending on the question that's being asked. So for example, PBPK tends to find extensive use based on the level of regulatory precedent and experience that exists in questions for helping address questions like DDI and the impact of concomitant medications on the investigational drug. Where it's more challenging or perhaps needs a little bit of additional clinical data to support that would be situations like understanding the impact of food effects. QSP and certain applications of PBPK in particular, to date, have been used to inform clinical program development at the sponsor's end. But quite often, given in certain types of applications, their limited experience will need clinical trial data in order to fully sort of convince reviewers of their applicability. So the limitations, I would have to say, depend on the types of questions. By and large, the agency has been very flexible. But if the person who raised the question would like to clarify a certain context, I'm happy to speak to that more specifically.
Great, thank you, Krithika. Miriam, can you talk a little bit about some of the commonly used PK software and tools used in MIDD programs for dose optimization that are acceptable to the FDA?
Yes, so there are multiple. We use a lot of non-mem R. There's Phoenix. There are really lots of different softwares, and it's really great to see that the FDA is also more and more open to open source software such as R.
Great. Looks like we've got one final question. I'm going to pose this to you, Miriam. Someone says, it's my understanding that the see if this makes sense to you. The mean concentration data is used, and then simulation up to 100 subjects. Is that correct?
Yes, we can do that. We use the mean, and then we use the variability of patient population covariates and of the respective populations, and then we can simulate up the population. Yes, I hope that answers the question.
I think so. If not, they'll get back in touch with us. Awesome. Thank you so much, Miriam and Krithika, and thank you to our audience for their attention and those excellent questions. Oh, it looks like we've got one more that came in. For dose optimization, under what condition could you select the higher dose level as the optimal dose with similar efficacy and safety for 2 dose levels? Is it possible to argue with the FDA if there's a trend but no statistical significant difference?
Yeah, this is an Optimus question more than an MIDD one, but I'll take it. So the answer to that question is it would be very difficult. If there's truly no difference between the 2 doses, not just on an overall response basis. So the secret here, or the intent here, is not necessarily just the ORR, but also looking at things like depth of response, time to response, duration of response. If there's truly no difference in the observed data after a randomized study, for a randomized dose finding study between those 2 dose levels, there will be very little rationale to pick the higher dose level to move forward. But yeah, we're certainly happy to, as I said, if there's more specifics around the context, we're more happy to provide a specific response. Short answer is it's hard.
Okay, great. This might be a question for Miriam. I'm not sure if I quite understand this. I think what they're saying is, if the drug is a small versus a large molecule, how does that affect POP-PK PD models?
We would apply very similar models, but then of course we need to adjust based on if it's a small or large molecule. There are separate things we can include in the model to address those factors. So this is also quite an extensive answer, basically. So happy to follow up if there are more questions, but we can address for this and in our models.
Great. I think this is the last question. Folks are obviously enthusiastic about this topic. When you're performing simulations, I think this is for Miriam, the original data set for the model building has to include subjects with similar intrinsic and extrinsic characteristics as the subjects to be simulated. Is that correct?
It depends. It depends if these covariates were included in the model. Ideally, yes, similar, but we can also test for differences in those simulations and then test actually how different the new population is from the original population used. So it's not entirely that we have to use very similar covariates. It really depends how the initial model was developed and what was included in the model.
Okay, fantastic. Thank you so much to our speakers, Dr. Traim and Dr. Shetty, for this excellent presentation. Thank you to our audience. We've got a few announcements and then we're going to wrap things up. Just want to let everybody know that our next webinar on the series will be on September 13th, when Certara Vice President of Toxicology, Dr. Jim Herman, will present an eChalk talk with AAPS titled FDA Modernization Act. What does it mean for pharmaceutical scientists? You can register for this upcoming webinar on the Certara website. Please do remember to take the survey that's going to launch as soon as we end this webinar so that you can share your important feedback with us. Just want to thank everybody on behalf of Certara. Thanks for attending today's presentation. This concludes the webinar. Goodbye and have a great day.